Okay, so good morning. Welcome to my INI 332 course project presentation. And Muhammad Sajid this side doing MTech at Sense. So I'll be talking about PCR specifically. Okay, so I'll be talking about PCR. Specifically, the topic is demystifying PCR test challenges, alternatives in the future. So I'll be reviewing this. I'll be focusing on COVID as well as fungal infections. Yeah. So why this topic? Because we are all aware that this pandemic hit us very badly, and nowadays every day there will be some news headlines mentioning about PCR, and so my intention was to understand about PCR in detail and share the understanding with you all. And I also want to know if it is an interesting topic to pursue in future. So here is a collage that I just made with this news headlines, and left side there is a COVID negative certificate that we all, I mean, almost all of us have currently. So what's the outline of my presentation? So today I'll be discussing about these topics as in given here. So we'll follow the procedure of just of a scientific paper. So we'll start with abstract, then go to introduction, the principles of then RT-PCR, quantity PCR, inhibitors, and move to COVID and fungal infections, and end with this diagnosis of human fungal infections, and then reference. Yeah. So let's get started about abstract. The polymerase chain reaction is actually a potent biological tools ever built. We are already aware of that, and it has different applications from medical diagnosis, biotechnology, forensic science, and food quality testing, everything, even in astrophysics. Actually, in astrophysics, the application is they are detected in the direction of dark matter using this PCR associated technique. If anyone interested, I can share you the paper. And the PCR is also modified to accommodate different needs. Now, like from RT-PCR and PCR. So this immense application of this PCR test and current health crisis we have faced is made it essential to understand and educate people about all these principles, mechanism, limitations, and alternatives. So what is the scope of my presentation? So I will be discussing about the different aspects. My main idea is to give an overview. I'm not planning to go into any topic in detail. I am giving an overview, including history, principle, challenges, and this advancement in alternatives. We'll also look into the PCR-based detection of fungal infections, considering the India's current tension led to the spreading of the black fungus. So it has been around 35 years after PCR has been developed. This guy is smiling at the right side of my corner of this slide is an American biochemist who is Caribang Mullis, and he was working in CETUS Corporation that time. His story is really interesting. I will just mention a few lines about that. The story, so he was actually taking, people say like this, the story is actually like he was taking a night ride in his bike in a long highway near the Pacific Ocean, and he was thinking about his job. Suddenly the idea of the PCR struck his mind, and it is reported that before before the journey get over, he started smelling a Nobel Prize, possible Nobel Prize, and he have an interesting history. Actually, after this PCR technique developed, what he did is he started a company. He founded a company which sells jewelry items, where his locket were in the locket he included the amplified DNA from the celebrities. That kind of that's an interesting story. So he was awarded with the Nobel Prize in Chemistry in 1993. So that's about Caribang Mullis and history. And actually, this COVID-19 spread as a global pandemic has multiplied this usage of this technique. So where we do, if someone asks where we do PCR, like everything in this plant has a DNA or RNA in the genome. So if you want to detect anything, then you go with PCR. That is the power of this technique. And apart from the medical diagnostics like COVID detection and this tuberculosis detection, actually WHO has mandated PCR for tuberculosis detection, and it has applications forensic science like finding the killer from these body particles and all. Then food quality testing. Then to do the human evolution, you can go and check the DNAs of mummies in Egypt and pyramids and all. So as I said, it is also proposed for potential for dark matter detection in. It's actually, that paper is actually still in arXiv, but if I want, I can send you that. And India is also currently facing a higher infection rate of this black fungus, which is it is actually, its name is mucormycosis, and this white and yellow fungus are also reported. So we will look in briefly into all these topics to give an overview. And in 1991, the Roche, which is we know that the pharmaceutical company actually has bought this technique, and he has started using for medical diagnostics. So before jumping into PCR, let me tell you about the basics of a DNA. I know you guys already know about it, but I will just mention as I am more into basics than going to advanced. So this DNA, which is also known as the deoxyribonucleic acid, is a molecule that is actually that coil around each other. You can see that coil around each other to form a double helix. Then the DNA strands are known as a polynucleotide, as they are composed of a simpler monomeric units, which have this nitrogen-based adenine, thymine, guanine, cytosine, and it is a, I think it is in RNA, then it is uracil something. Then when we'll have a sugar called deoxyribose and a phosphate group, that is the basic structure of the DNA. We'll give the direction from 5' to 3' and 3' to 5', and we can see there are a lot of hydrogen bonds. So in two lines, polymerase chain reaction is actually a that is an interesting fact. So in PCR, actually, we are using this. We are usually use these terms. I will just explain what all this. So the primers are in the primer. Actually, the synthetic strands of about around 80 to 25 nucleotides, which is complementary to the DNA target sequence we want to amplify. And actually, two primers will be designed in PCR. There will be a forward primer, and there will be a forward primer, and there will be a reverse primer. And so one is in 3 point directions. And then forward primer, then forward primer and reverse primer will start synthesis with the help of a DNA, with the help of the enzyme. And there will be some magnesium because it is acts as a cofactor for that enzyme. And definitely there are nucleotides that get stitched to the DNA to make that DNA double helix structure. And so the principles of PCR technique, these are mainly the three techniques in the PCR. One is denaturation, another is annealing, and another is elongation. So all three is known as a one cycle. So in denaturation, what we do is we will increase the temperature to around 90 degrees. So as you see in the previous slide, there are some hydrogen bonds. Primers will get attached to the strands, and then in the elongation or extension step, the DNA polymerase enzyme extends the primer using this deoxyribophosphate. Then actually this is a thermostable DNA polymerase because since there is we have changed the temperature like 90, 50 like that. So it is an important that our enzyme we are using as a thermostable. And here you can see a picture of how this doubling or the 2 power n multiplication of the DNA happens. This is usually represented by the equation 2 raised to n. N is the number of cycle, and actually the equation go like this: P equal to T into 1 plus E raised to n, where E is the efficiency, and for 2 raised to n we assume that efficiency is 100%, and we are getting the 2 raised to n equation, and the P is the amount we measured after n cycles or the product. T is the amount of cDNA from this. If you are using RT-PCR, then after reaction, then E is the efficiency. And one thing I mentioned that I have for the reference of this picture, I have mentioned here, and in some slides I didn't mention, but that is it. That can be found in my reference section. So you can see the number is drastic for each cycle. You can see that the amount of DNA is drastically increasing, and it is going a large number. So that is the beauty of this technique. And so the DNA polymerase, that is the enzyme we have used, which is actually responsible for the copying the sequence starting from the primer and form the things. Actually, we chose a stack which was actually isolated from the thermal spring in Yellowstone National Park, and the enzyme is important. It is heat tolerant, so we can both it. You can use it in PCR because we are like you see, you can this temperature is varying like this, so it is important we have a thermostable DNA. Enzyme. So that is about PCR. Let's go to RT-PCR. So in RT-PCR, in the PCR we usually have a DNA as a raw material, but in RT-PCR we have a RNA as a raw material, and after that we will make a complementary cDNA from the messenger RNA. Then we will amplify this DNA using the common PCR technique. So the advantage is like when RNA is usually degradable, easily degrade, but cDNA will have a long lifetime. And some challenges we feature is like these mRNA levels are actually fluctuating in the different tissues, and RT-PCR is not it be effective towards a crowd of patients. And the picture you are seeing is a thermocycler where we change the temperature. This was the one which was developed by the CITES Corporation, and this is the modern one. And yeah, so let's go to real-time PCR. So in the RT-PCR, we are in the end product we analyze using the gel electrophoresis at the end in the olden times. Now we are used to we are using this fluorescent dyes. What the health thing is like we do not require this gel electrophoresis to analyze the data, which will reduce the time. So the probabilities for contamination are also significantly less, as everything is happens inside a closed tube system, and the post amplification this thing is eliminated. So that's the interesting thing. So let me tell you how this fluorescent dyes works. I think Mayank has already mentioned this, but I will also mention anyway. Prepare the slides. Okay, so this is the thing. This primer we have. We'll have a reporter of fluorescence here and a conjurer of fluorescence. And the thing is like this is just these dots are not just from this slide design and not related to with this. And this they will start emitting fluorescence. They will start emitting. There will be some fluorescence comes out. So it will get, as you said, it will get attached to the RNA part, and after that this enzyme starts synthesizing. I can see in the synthesizing, and after that when it reaches there, this gets separated, and R gets separated, and R will show the fluorescence. So that will be analyzed using a setup like this. So all we'll have a lot of this will arranged in this one. And for each one the fluorescence will be emitted, that we will be detecting using a CCD camera. So yeah, that's about the fluorescence. And right, so we have a lot of complications in a PCR technique. It's not easy as it looks. So there are inhibitors which I will be explaining in the next slide, and GC rich templates, long templates, error. This is a multi-step process, so there are a lot of possibility of errors, and non-specific products may also come. Yeah. And so let's go to PCR inhibitors. PCR inhibitors actually the chemical substance that is present in analyzing the sample and reduce the sensitivity to PCR actually. So these are some examples of inhibitors. For example, urea, salt, phenol, ethanol, and all this will reduce the specificity. So how it actually do is, for example, if you have the urea, it will degrade the DNA polymerase. If they have phenols, it will cross-link with the RNA and cause trouble in RNA extraction. That kind of stuff is there. So we had to take care of these PCR inhibitors when we are designing things, and that's a challenge actually. Nowadays we are going into the nano PCR. I mean, we are trying to miniaturize these machines and all. So that times it is a bigger challenge to take care of these PCR inhibitors. So let's go think about the challenges and let's discuss about the challenges and future. So we know the PCR technique is relatively costly. As Mang mentioned, we need a thermocycler, agar gel diffusion array, the olden time. The other chemicals. We also need a trained staff and adequate space with a lab setup. And like the chemicals are also really expensive. And the transport. When there's a pandemic like this, we need to get the result at the earliest as possible. When it gets late, it will become more dangerous. I mean, that is become dangerous. So the transfer to the laboratory has actually increased the turnaround time, and that's also an issue. And the low frequency mutations in DNA is actually undetectable using PCR, and people say PCR is not quantitative enough. What we, as I said, in the future we wanted to miniaturize the PCR devices. Instead of a lab setup where there is a big thermocycler sitting there and some trained staff is doing all this stuff, and it takes a lot of time and money and effort. But we are trying to make some PCR machines which are set. I mean, this detection method using PCR, which are very small. Nowadays, if you go to a hospital, hospital or a simple, everyone have a thermometer with them to detect the temperature. So at the same way, we will also have a PCR strip or something along with that in the future. That is the future we wanted to see. And the conventional PCR also have the issues, as I said, the massive size and the heat loss, high thermal inertia, power loss, and all kind of issues are there. And also the RNA extraction kits, swabs, lysis buffer, the qPCR machines, all under scarcity when there is a current pandemic situation like this. This technique will also show errors due to the multi-stage process that required for the extraction of the DNA. And as I said, it can also give wrong results due to low viral load in some patients, and it can sometimes give false positive results because the test may cross react with something other than the COVID virus or the virus we want to detect. And there are also chance of laboratory errors like the clerical error or testing a wrong sample or cross contamination. There are a lot of issues like that, and some of the people have come up with the solutions, which is interesting. Actually, in the PCR, one of the challenges Mayank mentioned is the temperature variation. We had to change the temperature, increase, decrease, increase, like that. We had to go. But in the recently, a professor who is locally professor of bioengineering at California, Berkeley, he devised he has made a setup using the plasmonics, which is a mini kind of PCR, mini PCR, you can say like that. So he was able to heat a 120-minute nanogod film, which is in contact with the DNA solution microfluidics well. The mechanism allowed him to do the 30 times in 5 minutes. So, and also in the future of PCR, the importance of microfluidics techniques are very much bigger because I will explain later. So, and also in the companies like Quantum DX or other companies have developed a lot of this benchtop devices which can reduce the test as little as 7 minutes, and it has improved the sensitivity under the 1 cell per ml to 150 cell per ml, which has used with the microfluidics, as I mentioned, microfluidics and the nanowire field effect transistors. I think these things are already mentioned by Calvin yesterday, I guess. And the large diagnostic laboratories, we use this, automated robotic things. So the error is very much less. And if there is a family of, big family of viruses, people have designed heterogeneity primers to detect this specific family of viruses. And there are also come with the Merck curve analysis and that kind of statistical techniques to detect the multiple pathogens. And we also have these multiple assays which help like detect, do things together. And there are also quantity PCR methods developed, which as I mentioned, there are like cybergreen and evagreen to measure the dyes, which dyes like cybergreen and evagreen to measure the amplified DNA in real-time quantitatively. And nowadays the sensitivity and specificity are drastically increased because we are able to use dyes and we have improved our instrumentation. Nowadays everything happens in a closed container, and the possibility of something coming from outside is really less, so contamination will be less. And also there are some methods, safer sequence, which is recently published in Nature, I think maybe in June, so which is able to detect the low frequency mutation DNA using PCR. And since it's a multi-stage process, I said there are a lot of possibility of error. So there are some normalization strategies which are mentioned by these scientists to reduce the reduce the error in real-time PCR. So in the future, we will see is in few years we'll have handheld PCR devices or POC devices which stand along with the thermometer, as I mentioned. That's the future we wanted to see. So let's go to the COVID cases. Let's go to COVID. Nowadays we are using this RT-PCR for extensively for COVID detection, and it actually helped us a lot to help us fight the pandemic. If Carrie Mullis was not able to come up with this idea, I don't know what we will be doing now. I really wonder about that. And actually this DNA nucleotide has RNA with around 30,000 nucleotides length, and which have these 27 proteins, include the structural proteins outside. Actually, these S genes are responsible for the receptor binding spike proteins, which enables a virus to infect the cells. And you can see that these are the target genes in COVID. These are the main target genes, and these are the primers, and we do that. So what actually we do is, if I want to do PCR, what I will do is I will find a specific target gene in that that virus or bacteria, whatever I want to detect, and I will use the tool. For example, you have Primer 3 software tools where you can design the primers. We can design the primer sequence based on the target gene. If you give the target gene, you can make it. You can get the primer sequence, and you can send to some companies which you already, who make these things in a commercial scale, and you will get it done. That's the way we have to do it. So the COVID detection is all aware. We had to sample thickness. We will take a few millimeter from this novel, lower respiratory tract, and which like this, and that is the viral transportation medium. And then we'll use the heating or chemical lysin to inactivate the virus, and the viral DNA then purified using magnetic beads or the column-based RNA purification kits. Then we will do the PCR. The amplified product will be tested using Tachman probe fluorescence or DNA intercalatin dyes, and the specific threshold of cycle is set to differentiate between positive and negative result. In India, it is around 35. So what that means is, if you are able to see enough fluorescence before 35, that means you are COVID positive. And if you are not able to see the enough fluorescence even after 35, that means you are COVID negative. So that is the way it works. And the limitation is that threshold cycle value may change depending on the specimen collection, the transportation, time gap between the specimen collected and the testing, and the analysis, model of thermocycler, even the model of thermocycler, the PCR system we have. Depend on all this, the threshold cycle will vary. That is also a challenge that we have to take care of. So if you reduce the cycle, instead of ICMR suggested 35 cycles for the threshold cycle, if you reduce it, what will happen is you will detect the false positive result, because if a little contamination can give false positive result. And if you increase the high cycle, then that will leave the COVID case undetected, resulting in high false negative result. So we have to take care of. But this cycle is vary with the country. I see that in US and UK, the cycle threshold is different. That they have set up depend on the condition, the state, and all. So the COVID detection, there are a lot of other alternatives than this our PCR method. CRISPR-based detection and isothermal amplification are already well explained by Mayank, and that's great. It was great. And actually, the isothermal amplification, the technique does not need a thermocycling. As you mentioned, we have a LAMP and RPA, but like loop-mediated isothermal amplification and recombinase polymerase amplification. So like a strand displacing DNA polymerase initiate the synthesis in there, and the other primers will form the loop structure to facilitate the subsequent also amplification. So that is an interesting thing. And nowadays we have the sequencing based diagnostics. Nanopore is one of the interesting thing that is doing help us with the sequencing based diagnosis. What we do is we will just send that the DNA or whatever we want to sequence, we will just send through the nanopore, and depending on the change in the we'll send through the nanopore, and depending on the current and all, we'll be able to we'll measure the current and all, and depending on changes, we'll be able to see what is actually moving through this nanopore. Is it a TGC or like that. So nowadays we also have the sound-based COVID detection. I think IAC has a Coursera project where we're hearing from the sound of a COVID patient, we can be able to differentiate between COVID positive and negative cases. As you know, the COVID mostly affect lungs, and there will be some changes in the sound-based thing. Then comes the digital PCR. So what we'll do is we will partition the PCR solution using a microwell plates into thousands of nano droplets. We'll have like this in the machine. We'll separate this to nano droplets, and then we will separate to PCR reaction happen each one of them. Then sample will be checked for the fluorescence readout. And the DNA nano switches. In the DNA nano switches, we'll have this nanoswitch oligomers bind the viral RNA to undergo a conformational changes, which can be visualized in agarose gel and all. And we'll also have this antibody detection method, IgG, a kind of antibody detection method that Mayank also mentioned. And people say that the computed tomography or CT scanning can also help us to detect this. And so BCR-based diagnosis of human fungal infection is also there. As you are aware, this mucormycosis, these things are there, and like black fungus is there, and it actually affects our eyes. And WHO guidelines in 2019 guidelines, they say that if you are affected with black fungus and you are not in a stable state, you will have to remove the eye or whatever the body part, and that is a tragic things about that. So we need to detect at the earliest as possible. Otherwise, if you get late, I mean, your eyes will go on. That is really bad. So unlike the viruses, doing PCR testing in fungal samples is little challenging. Actually, the fungi cell walls embed the lysoorganism, and hence the extraction of DNA is little complex, and this may result to the false negative result. And also fungi spores are ubiquitous environment. It is everywhere. So there will be contamination. Easy contamination will be there. And the conventionally we'll use this fungal. For the fungus detection, we use this antigen testing, radiological imaging, there's a microscope examination, etc. And but the issue is don't have a specificity and sensitivity compared to PCR, and is also take a reasonable time. I was tried to actually, I wanted to show you the target genes and all, but there are a lot of complexity associated with it, and I thought I will not do it. I want to make it simpler. And actually, recently the Bruker have launched a real-time PCR for invasive fungal diseases, Fungi Plus, and like this technique, this product to detect this Arpagillus and Candida DNA respectively. And this is the bibliography I have used. And thank you for listening. Before that, let me conclude. I just forgot to conclude. Okay. Yeah. So considering the current situation, it is very vital to develop a rapid, accurate, easy to use, and less costly diagnostics, especially when there is a situation like this pandemic or like COVID or Zika virus or whatever. So even though we have the advanced variants of PCR like digital PCR, those kind of things, but all the labs in our country are not still equipped with all these instruments and experts. So we need to equip the government and private testing facilities with advanced PCR methods like the microfluidic PCR methods or the digital PCR and all. So the future research in this area will be on miniaturizing PCR devices, and branches may like nano PCR that may arise, and that is an exciting future I'm seeing in this. And we had also focus on growing more experts and do R&D in this field, so that we'll be ready to fight whatever the troubles the future bring us. And there is some online tools you can simulate the PCR. You can design, if you give the sequence, you can design the primer using this. And if you have designed the primer, you can send to companies like this for officiated making this. And I just added the link to like the approved PCR kits manufactured by Government of India. And if anyone wanted this slide, please mail me here. I'll be happy to share. And let's yeah. Any questions? Yeah, that will be. Thank you.
Thank you, Sajid. It was a good presentation. So what you are saying is that the major challenge or something like that for future is basically instrumentation, right? So making it smaller and then maybe the novel methods, incorporating that and so on. Hello?
Yeah, yes, sir. Yes, sir. It will be a major challenge.
Okay. Yeah, that's true. So I had one question. So you mentioned about this, this astrophysics application. I was just curious.
Yeah.
How do they use that? Yeah. I mean, just a very brief thing. Yeah.
Oh, okay. The idea is they have made a long road like structure, and they will just attach some DNA or RNA strands on that in a row, and depend on the movement of the dark matter. There will be some change in the space or some friction or force may be there, and it will cause the DNA or RNA to fall down, and that can be detected using this PCR amplification, and based on that we can say the direction of dark matter movement. I can send you the paper, sir. I will send you the paper.
Okay, all right. Yeah. Okay, yeah, no. Okay, so thank you. Thanks for the presentation. So we will meet tomorrow at 10 o'clock. One more, I forgot to mention one thing, which is that these presentations can be uploaded in the